are going to get started. All right, there are 6 sections of Bio 93 this fall, and so to be in this class right now, you should be enrolled in Section B, 1 to 150, with O'Dowd and Edinger. If you're not enrolled, but you want to be in this class because you're trying to move around, you have to enroll through WebReg. And if that doesn't work, you can always contact the counselor in BioSci Student Affairs, but we as faculty in this large class do not handle any adds or drops. Okay, and then I also need to tell you, and I don't see the guy that's filming, that this class is going to be filmed throughout the quarter. And it's going to be used, posted on the UCI website as an open course. And so I need to tell you that we have anybody who participates in an activity that we do on stage will need to sign a release saying that they're okay with being filmed. Most of the filming is going to be of me, but there may be sometimes filming of the audience, not very much. And if you don't want to be filmed, make sure you sit in the second half of the class. And I'll give you some more information on this on Monday. Okay. Okay, I can't move forward with this for some reason. Moving forward. This is my great pleasure to welcome you to Bio 93. And first I want to know how many of you out there are freshmen. Raise your hands. Okay, great, a full class of freshmen. So welcome to UCI. How many of you are from outside the state of California? Raise your hands. Okay, we actually have a number of people. I would like to say that the only good thing I've seen about this downturn in the Californian economy is that we have started enrolling more students from outside the state of California. And I think that that geographic diversity is really enriching to the student body as a whole and creates a much more diverse and enriched environment. So I welcome all of you here. Today, you're going to be starting on Bio 93, which is the first class in the core required for all biology majors. And we hope this is the start of a fantastic 4-year journey where you're going to learn a lot of really interesting biology. In addition to that, we really hope that you take the advantage and the opportunity to explore new ideas, develop your critical thinking skills, and really become an active participant in the larger learning community at UCI. Because it's those activities in aggregate, not just the biology that you'll learn, which will make you successful at meeting the really exciting challenges that you're going to face as our future healthcare professionals and educators. There will also be some of you in the class that decide after this class that biology really isn't for you. But what we hope you get out of this class is a foundation that allows you to make informed decisions going forward in your life about your healthcare for yourself and for your loved ones. My automatic clicker has let me down, so I'll have to go over here. This is a team taught class. I will be teaching this class with Dr. Amy Edinger. My name is Diane O'Dowd, and we are both professors in developmental and cell biology. I will teach most of the lectures in the first half, and Dr. Edinger will teach most of the lectures in the second half. But we work together to put this class together, and we will work together throughout the quarter in terms of assessing how activities are going, how your performance is going, what we need to do to help you learn the material in the best way possible. I keep clicking forward. I might have to stay over here more. Okay, and before we go into the biology and administration, we wanted to give you a little bit of background about ourselves. And so, by way of background from me, I grew up in a small town in southeastern Michigan, and I started my biology career 37 years ago, almost to the day, when I flew from Michigan to Northern California, and I arrived as a pre-med student at Stanford. And that's how I started. And after, by the end of the first year, I had gotten involved in research in the laboratory of a neurochemist, Dr. Larry Yang. And I quickly found out that that was my calling. I really liked being in the lab, doing experiments, solving problems. And so I stayed in his lab for 3 years, did my honors thesis in neurobiology. And then I went down to UCSD, where I did my graduate work. And that's where I first became really interested in how neurons in our brains communicate with each other using both chemical and electrical signals. And those are studies I still carry on today. I then went back up to Stanford for a postdoc and came back down here in 1989 when I started as an assistant professor, and I've been here ever since. Okay, so in addition to our really important work in teaching you guys in the life of a faculty member at a research institution, we also conduct full-time research. And I have a laboratory in which I have a postdoc, a graduate student, and 3 undergraduates right now, and we're all working on some aspects of the cellular mechanisms of learning and memory. And we do most of these studies in these tiny little guys, fruit flies, Drosophila melanogaster. While you might find it hard to believe that these creatures can actually learn and remember information, they do so in a fashion that is very similar, not only to these much bigger, hairier animals here, but in fact, very similar to how you guys learn information, and myself. Okay, and so by way of an example, we can teach fruit flies to avoid a neutral odor by pairing this odor with a suitably nasty stimulus like a shock. And the most interesting thing is, when the odor and shock are paired, that it's the timing of the pairing that influences how long the organism remembers that information. So if there are 10 pairings of odor and shock, and there's very little spacing in between the pairings, then the animal will remember for the next few hours to a day. However, if you put 10 minutes in between each odor and shock pairing, they remember for the rest of their lives. Now, they don't live all that long, but it is the same principle that when they cram, their information is only short term, without rest periods in between. And that is really a message I want to leave with you. We put many mechanisms in place in this class to get you to keep up with the material on a regular basis, because that is the way you're going to learn the information for the long term. And that's really important in this class, because this class is the foundation for all the classes you will take as you go through the biology core. And if you learn it now, it's going to be a lot easier when you need to go learn it and build on it in the next classes. Okay, and so now I'm going to turn you over to Dr. Edinger.
Okay, so as Diane said, I'm also in the Department of Developmental and Cell Biology, and I started out my life in San Diego. I grew up there, and I went away for the first time when I went to UC Davis for school. I always wanted to be a veterinarian, probably because my mother hated animals. That's why I ended up at Davis. And while I was there, I needed to volunteer with a veterinarian. And there were so many pre-vets at Davis, the only way I could manage that was by working in a research lab. Nobody in my family had ever been involved in laboratory science, but I decided to give it a try. I did a 199. That's research for units, which you can do at UC, and I encourage you guys to think about it. And I discovered that I really love doing research, so much that I ended up doing a combined degree, both a vet degree and a Ph.D. And I did that at the University of Pennsylvania, which is in Philadelphia. I lived there for 12 years doing a vet degree, a Ph.D., and a postdoctoral fellowship. Then I finally got to come back to Southern California, and I came to UCI in 2005 and started my own research lab. Right now I have 3 postdocs, 2 graduate students, which happen to be some of your TAs, and 4 undergraduates and a research technician. So it's gotten to be a pretty big lab and takes a fair amount of time in addition to the important work of teaching you guys. So I mentioned that we're a cancer biology lab. What we're trying to do is develop new cancer therapies that selectively kill cancer cells by starving them to death. And the way we're trying to do that is preventing them from getting at nutrients. So here I'm showing you some immunofluorescence microscopy. It's stained for amino acid transporters in red. So over here, the transporter is on the surface of the cell. And in green, I'm showing you the trans Golgi, which you will hear more about shortly. So here's the trans Golgi. And this big black spot in the center, what do you guys think that is? Starts with an N. Nucleus. Yeah, it's just not stained, but it's blocking the light. Okay, so this is a HeLa cell from Henrietta Lacks, a woman you may have heard of. She provided the sample, the cervical cancer sample, that became the first human cell line that could be propagated in the lab. All right, so here's a happy cell. When we treat it with our hopefully cancer drug, you see that the pattern of staining changes significantly. The transporters come off the surface and they're in these dots inside the cell, and the trans Golgi fragments. So our drug dramatically affects the intracellular trafficking of these molecules. And I picked out this picture in particular because one of my 199 students took it, an undergrad who is very dedicated and talented, took these images, which we hope will be in a publication soon. Now, last piece of my history I wanted to share with you is when I was at Davis, I did the Education Abroad program, and I went to England for a year. I was pre-vet, maybe pre-med, and I went into the office and told them that, and they were very discouraging about doing a year abroad, but I was very stubborn, and I still am, and I did it anyway, and I have to say it was one of the most valuable experiences of my life. It changed the person I am. I got to take classes with 4 other people and the professor, small labs. I traveled a ton. I met people from everywhere and got to go visit them in their homes. It was just a great experience. Here, they're very supportive of pre-professional people doing education abroad, and I encourage you guys to look into it. So I am looking forward to teaching you in the second half, and I will see you guys around this quarter.
All right, I want to echo what Dr. Edinger said. I actually did not take advantage of education abroad when I was an undergraduate because, in fact, we were told, nope, sorry, you can't do that because it just won't fit in with your curriculum. The first time I ever did education abroad was 5 years ago with students from UCI, and I went to Chile, and we were all involved in an education abroad summer program there, and it was, again, a life-changing experience. It was wonderful. Okay, in addition to my interest in learning and memory in fruit flies and the underlying mechanisms, I'm really interested in how people learn information, and very specifically, how students in a large introductory biology class learn information. So with funds supported by the Howard Hughes Medical Institute, I started a program about 6 years ago to develop new tools for improving the quality of instruction in a large classroom like this, and then assessing how those tools work in terms of changing your learning outcomes and satisfaction with how we're delivering the material. So you are our test subjects, and we collect data to evaluate the effectiveness of new tools, and we collect data by looking at how you performed on particular questions that we've given you or the surveys. And this is all within the context of the class. We're not doing anything in the class that we wouldn't normally do. This study has been approved by the Human Review Board here at UCI, and the study information sheets are posted on the website. Anybody who wants to opt out of this study is fully, it's okay to do so. It will in no way influence your grade or what you do in this class. Everything, like I said, that we do in this class, we choose some of these things to use for data, but they're all just elements in the class. But if you're under 18 years old, or you don't want your data included, just tell us. That's by writing to this person, Dr. Lei Sun, at this UCI email address. She will keep this information confidential until after the class. When we are coding data, we take out your names and we put numbers in their place, then your data will be excluded from the study. Okay? All students in this class are required to attend a discussion. These meet 1 hour per week, and they're a really important part of this class because they are the place where you get an opportunity to take the information that you learn here and have started to practice it a little bit and use it to practice solving problems in small groups in a mentored setting. And we have a group of fantastic TAs that are associated with running these discussions. Can the TAs stand up? I think they're all in back. There we are, up and back. So if you look, turn around and look back, there are 5 TAs that are associated with this discussion. And how many of you have met your discussion leader? Oh, good, a lot of you have. Okay, so anybody who had discussion on Thursday or Friday morning will have met that. So thanks TAs, they're going to do a fantastic job. They will not only help you learn the biology, but they're resources for all sorts of things about how you might get involved in research, study tips, all of those kinds of things. They are an amazing resource for. Okay, on the pre-class syllabus quiz, the most commonly incorrectly answered question was, if I have an administrative question, I should email the professor, true or false? And as most people said, 60% of the people said false, but 40% true. So the answer is false. In fact, we set up a message board on EEE, which is called an administrative forum, and it's a place where you can post questions, including those about clickers and online quizzes. And to be honest, it does cut down on the number of emails that the faculty have to answer, and there are 450 of you here and 450 in the class previous to you. But way more importantly is, if one of you has this question, there's another 10 to 100 that have the same question. If you post it on the forum, then either another student will answer, or if we answer, the faculty or the TAs, then that answer will benefit everybody rather than having it only go to one person. And we really try not to do mass emails only when things are extremely important and urgent in time. Everything will be posted on the message board so you can go get the information when you want it and need it. Okay, we also have biology unit forums, and this is where you can post questions about the content of lecture, where you're confused about how something works or you want to get some additional information about something. That's a place to post questions. And either the TAs, other students, or the faculty will answer those. New this year, we're going to be using piazza.com, which is a message board that we used this summer in the online Bio 93 class that I did. And it was really a good place to practice solving exam problems in an environment where lots of students could be working on the same problem. And I just don't know if it's going to work in this environment because there's so many people. So we're going to try it. We will give you an email. We'll probably start it about next week. And we'll give you an email, and this is one of our pilots. You guys are the first ones that we're going to try it on. And we need a lot of feedback from you about whether it's working or not. And so this is completely free. You will get the email that will tell you how to go and sign up, and then we will try that next week. And you can ask questions anytime during my lecture now or any other time in class. All right, we as faculty would like to be able to interact with each of you individually, but as you can see in a class this large, that's relatively difficult in a classroom. However, we do hold weekly office hours and problem-solving sessions where we will interact with the students that come and try and, from your input, develop questions and help you work on problems that are going to help you increase your understanding of the material. These will be Mondays from 2 to 4 in 1202 Nat Sci 1, which is just around the Ring Mall. And I will do the office hours in the first half of the class, and Dr. Edinger will do the office hours in the second half of the class at the same place and time. Okay, we do have email addresses, and you can use those. So the times when those are important is if you have a personal question. So a question that it has to do with health issues or other specific questions that aren't to be broadcast to the whole class. Or if you have asked questions in all the different formats that you can find and still didn't have your question answered, then you can email us and we'll try and help you with those. So we try and be really good about responding to emails as quickly as possible, but we respond more rapidly in some circumstances than others. And for example, if we get an email like this, we're a little bit less likely to respond straight away. Me, because it takes me a long time to read it. Came from a non-UCI email address, there was no subject, so I didn't know what it was about or if it was important. Has kind of a yo address that I'm not sure if it's to me or somebody else. Has a lot of shouting in it, and there's no name at the bottom. And text messaging, which really I'm not up on. I have to really think about it. So what we would like you to do if you email us is just use a simple professional format where you have on the subject, it comes from UCI email address, has Bio 93, and this would be saying section B, not section A. That just helps me identify who you guys are. Open with the proper salutation, dear Dr. or Professor O'Dowd or Edminger. Do use punctuation and reasonable grammar. Doesn't have to be perfect, but just use your regular English skills. Don't use text messaging or abbreviations, and close with your name. And the reason why we tell you this is because I learned from experience with my own kids who are just a little bit older than you. My daughter just finished college this year, is that they didn't know that these were the rules, and that when you use these to email another professor in another class, you use it to get information about the professional school you're applying to, a job you want to get. You will get a lot further with an email like this than you will with a much more informal email. And so it's to help you in using all your professional communication, written communications and email should follow this format. All right, now we have to talk about a little bit about course mechanics. We do, as I said, have a website that is, we have a number of things at that website. On the home page, we have important announcements, new information, that's where class files are posted. We have the syllabus, lecture schedule. Every once in a while, we'll have a change in readings, and if we do, we'll say on the front of the website, see updated lecture schedule, and then you can download a new updated version. For every lecture, we have a set of learning goals. If you ever come out of a lecture and think, well, did I learn what I was supposed to? You can go and look at that and see. You'll see what we expected you to learn from that lecture. There's also textbook information, what type of textbook, where it is, what are the advantages and disadvantages of the different versions of the textbook. And then this is where you'll be getting your lecture outlines. We'll be posting them generally 24 hours before the lecture, and then you can download those and bring them to class. So we recognize, in fact, that there are lots of diverse learning styles among you in class, and with those diverse learning styles come diverse note-taking habits as well. So I'm just going to go over this briefly. People that like to take their notes on paper, so paper note takers. Some people just come and bring, blank sheets of paper and they write on them, and that's perfectly reasonable. If you bring a printout of your PowerPoint outlines, and they are outlines, they do not contain all the information that you'll need, a lot of that will come in class, then we recommend printing 2 slides per page. I know it takes up a bit of extra paper, but the diagrams and the details of the diagrams are really important information. And so if I have something on a diagram that's in my lecture slides, you are responsible for knowing that. In your book, the diagrams often have way more detail. That is extra, but what's in the notes is expected that you know it. Okay. Some people like to bring laptops, so we had to start adding to this iPads and tablets to class, and so they'll download their lecture notes to that or just take notes on that. And that's okay. Make sure that you download your files ahead of time. And it's really important that you're respectful of the people around you and what's happening. And so don't use your electronic device for Facebook. Email, I have to cross that out. That was like 5 years ago. Playing Angry Birds, poker, whatever else. It really does you no good, and it doesn't do the people around you any good either. Okay, and the other thing I have to say, so one of our studies last year was to compare the performance of students who used laptops versus students who did not use laptops. So we had paper note takers and laptop note takers, and the results of the study said that those people who selected to use laptops performed slightly worse than predicted, and those that chose to take notes on paper performed slightly better than predicted. That's on average, doesn't mean it happens for everybody, but that's just important information to know, and we actually don't know why. We don't know the underlying reason, But one of our hypotheses is that some of the things that we're trying to teach you in here require you to learn information that's very visual, and drawing it and writing it often helps activate different parts of the brain than if you just listen to it and type. So that's, again, it's up to you. We want to make sure that this classroom accommodates a diverse variety of learning styles and note-taking styles. That said, we're going to have a laptop-free zone because also we learned last year that many people that take notes on paper are distracted by people that have laptops. And so, starting on Monday, we're going to have people that just want to take notes on paper fill in from the right-hand side of the class over here, and the people that want to use electronic devices will start over here. And you can be a back-of-the-room person or a front-of-the-room person. Just start filling in from this side if you're using electronic, and start filling in from this side if you're using paper. And that will accommodate, hopefully, everybody so they can do what they feel most comfortable with. All right, we have a number of goals in this class. By the end of this course, you should be able to explain basic concepts of cell and molecular biology using correct terminology, construct and critique logical arguments, accurately describe what you observe, and we'll give you some opportunities to do that. Solve problems during learning, including figuring out where to get information and how to evaluate it, and to work effectively in a team as you're solving problems. So we'll give you opportunities in the classroom to do that. And in this, with this as these as our common goals, we as faculty commit to making the lectures as interesting, relevant, and interactive as possible. So you as students can come to class and actually be participating and active, so your learning starts here rather than you just transcribing what we say in class and then go home to learn it in a non-mentored environment. We want your learning to start here in the classroom. Okay, to do this, we include a number of different things that we do in class. The first are clicker questions. And so those are questions where we're going to ask you in class, you're going to answer the question, and so I want you to take out your clickers right now and turn them on. And what happens when you turn them on? Yes, you get a little blue light. Good. Okay. And if you get a little blue light, and now you push button A, B, C, D, or E, what happens? You get a red light, right? And that's because my receiver is not receiving your signal. That's because I haven't turned it on. So now, if I activate my receiver, now if you click in, very good. Now your answer is being recorded. And you don't have an answer to anything because I didn't ask you any question, but you just click in something. So whatever you click in last, before I stop the recording, so I'm going to stop it now. Is recorded, and most of you are A people, okay? That's the grade that you are going to get in this class, right? Okay, so now let's move forward and actually answer a question. So which of the following events is taking place this evening? Here are your choices. And you can talk to your neighbors if you want, and I'll give you 30 seconds to click in on this. Okay, click in if you haven't. Stop, and we'll see what you said. Oh, very good. 88% of you got the correct answer, which is men's soccer is going to be at Anteater Stadium tonight. We go, my family goes a lot. We started going when our kids were really little and they were in soccer. You can go. And it's free if you have a UCI, and they're really fun to watch. The women are not in soccer here today, but they will be next week, so they're also great to watch. All the sports teams are really fun to watch. Okay, Madonna unfortunately is not in concert at the Bren Event Center. I put this in because actually Madonna and I were classmates in high school. That's one claim to fame is that she grew up in Rochester, Michigan, just like I did, and went to Adams High School. So the challenge for dodgeball world record was set by you guys. How many? 6,000. Yeah, very good. And target after hours. I thought this was actually the right answer, but I guess that was last night. Is that right? Okay. Okay. So you guys knew. I thought that was the right answer too. Okay. Very good. Okay. So I use clickers not to take attendance, not to try and get you here. What I use clickers for is to figure out what you know before I give a lecture or ask a question, what you learned after I ask the question, and how I might better help you learn the material. So you'll see when we give you points for clickers, it's very generous in the sense of you don't have to do it every day, you don't have to answer every question, but I really appreciate your participation in all of them that you can because it helps me be a better teacher. Okay, we also do class cards, and that's where we look at more open-ended questions. It's not an A, B, C, or D. And we work in small groups of 3 to 4. And so that's why I asked you to bring 3x5 cards today. And the first thing we're going to do is you're going to form a group, don't do it yet, with 3 or 4 people around you. And on one card, write all of your names. Introduce yourselves to each other, and then we'll use this for an activity. If you need a card, raise your hand, but somebody around you hopefully will, but we have cards from TAs, so raise your hands if you need them. Okay, so I see some people have introduced themselves. Hopefully you've met at least, okay, we need to quiet down. Hopefully you've met at least someone new. Part of why we're doing these cards is to get you to talk to each other about biology because learning biology is sort of like learning a foreign language, and you learn much better when you speak it. And the other reason is because many of you are new here, you don't know very many people, and so this is a mechanism where you don't have to say, oh, hey you, because their name is on the card and you can read it and actually maybe remember their names. And that's really important. Forming part of the learning community is a really important aspect. Okay, so we're going to use those cards in just a minute, so hang on to them. Okay, and then we do demonstrations. That's where we do physical demonstrations up at the front, and oftentimes we have students that come down and help with the demonstrations. Again, a different way to visualize the same material to help you learn the concepts that we want you to know. Okay, we also do pre-class quizzes. We have online lecture quizzes or exercises. We have online lecture quizzes. We have a pre-class quiz already for the syllabus. It was open for 24 hours this past day. And then that didn't have any points associated with it. It was just to get you used to using the quiz tool. The quizzes with points associated with them will start on Sunday. And the first one will be the quiz that will cover material that we're doing in lecture today, which is there's not very much material, as you will see. But this is to keep you up with the material on a daily basis. They'll be open again for 24 hours, and they'll cover the material of the lecture that you just had previously. We will also have online homeworks. There will be about 5 assignments, and the details will be presented in class about what we're going to do, when we're going to do them, and those will come up periodically throughout the quarter. Okay, and the grading rubric is on the syllabus, posted on the website. It tells you exactly how each of these elements is graded. Okay, and in that vein, we really want to stress that in this class and every class at UCI, academic integrity is really paramount to us accomplishing our mission. And that means that it really is important that you do your own work at all times, unless you are told to do work together. So our Bio 93 academic integrity pledges, I will use only my own clicker and complete all of my quizzes, exams, activities, and assignments without unauthorized assistance, and I'll not click in for anyone else, or provide, utilize unauthorized assistance to anyone else on quizzes, exams, activities, or assignments. And I recognize the value of academic integrity, agree to conduct myself according to this pledge, and signify this by clicking in A. And so, go ahead and click in. And we really do feel that this is important in this classroom where you're going to be taking tests under really stressful circumstances, and that you'll be sitting right next to the person, right, that we only have so many seats in the classroom. It's really important that you take this on as a personal responsibility. Okay. Okay, now I'm trying to go forward. And now I can't go forward at all. Okay. Let's see if that will work. No. Okay. Oh, okay, good. That helped. So did I go too forward? Okay. Every lecture outline will start with a page that has the lecture number and the title, as well as the reading material. And this topic of this class is biology, and most simply put, is the study of life. And while life is actually hard to define, we know living things by what they do. Living things emerge out of highly ordered structures like this sunflower. They reproduce using an inherited pattern that guides the growth and development of the offspring, so they have a characteristic of the species, like this macaque baby and its mother. They take energy from the environment. They respond to the environment, like this for a dragonfly that's going to get snapped up in a minute. And in this class, you're going to be learning lots of biological facts. And what we want you to do is start thinking about biology not just as a disconnected set of facts, which that's what it often seems like when you're learning something new, but in terms of the concepts. And one way to approach this is to think about biological systems in terms of their structural hierarchy. So to get some feedback about what you know about this so far, I want you to select the list that's correctly ranked from highest to lowest. And take just 30 seconds or so to do this, and you can talk to your neighbors. And then click in the answer. Okay, most of you have already answered, so click in if you haven't. And the answer is 97% of you are correct. The answer is organ, tissue, cell, molecule, and atom. So the guiding principle here is that the hierarchy that we're interested in this exercise is based on structural complexity. So the simplest units are at the bottom, and the more complex units are at the top. And it is interaction between components in each of these levels and between levels that lead to the emergent properties that we're going to be studying in this class. So how does a cell react and move in a different environment? That is because of how atoms in the cell interact, how these form molecules, and how these make up different components of the cell. Okay. So now I want you to do an exercise with your group that you formed on the cards. And this exercise is a little bit more challenging than the last one that I gave you, which is to put the following in order, highest to lowest, in terms of structural complexity. I'll give you 5 minutes to do this. Then you'll hand in your cards, and the TAs will collect them, because I forgot to do this last time. And then we'll establish the order as a class. And when I ask you for answers, you need to give me the logic behind it. And if you have any questions while you're doing this, raise your hands. Oh, okay. There's a really good question up here. good question, and the order that you put this in your hierarchy will depend on how you define that ostrich egg. Okay? So you go ahead and put that as your reason. Okay, so it has organs and all sorts of stuff in it. And what did you guys put next?
The heart.
Heart. And how do other people feel about that? Is that what they want to put next, or anybody else have anything? Heart. Okay. Heart. What type of, we did that hierarchy before. What type of, where does it fit in that hierarchy that we did when you did the clicker question? It's an organ. Okay, good. So what's next? Somebody in this section, tell me what you put next. Yes, way in the back. Muscle. Okay, anybody agree with that? Yep, okay, muscle. Okay. And now on the left-hand side, how about over here? What's next? Yes. Oh. Anybody here want to give me some group? Just tell me what you put next on the card. Yes, in the middle then.
Human sperm.
Human sperm, okay. Oh, somebody up here has one. Yes? Neuron. Neuron. Okay. Any others? Neuron or sperm, okay. So the person who says neuron, why did you say neuron?
It's a nerve cell.
Okay, neuron, nerve cell. The person who said sperm, what type of cell is that? Reproductive cell, it's a gamete, so they're both cells, right? Are they exactly the same? No, but you could argue long and hard about which one is more complex, right? Did you argue about that in your group? That's sort of what you should have been doing, is arguing about it. What would make one more or less complex? And in fact, if you were thinking about our hierarchy before, right, they could really be on the same line, right? Because they're both what? They're both cells, exactly. Okay, what would go next? Nucleus. Yes? Phospholipid. We have phospholipid or mitochondrion or nucleus. Okay? So what do nucleus and mitochondrion have in common? They're both organelles. Nucleus and mitochondrion both are surrounded by membranes. And what are membranes made out of? Phospholipids. So phospholipids would be just below this, right? In that they are many phospholipids contained within nucleus and mitochondria. at the bottom, we have hydrogen ion. Okay, so one more thing. We had a good question up there. Was there another item on this list that could have been put in a different location based on your interpretation? Ostrich egg was right? That could have been put another item. Muscle? Yeah, why?
It could be a tissue or a cell.
Right, that's a good point. The muscle was not identified here. It could be a tissue or a cell. How about another one? Think physics. Physics. Nucleus of... I'm a cell biologist. You know, a nucleus is in a cell. And when I wrote up this exercise the first time, that's all I thought it was. Until I gave this talk in front of a physicist who said, this is a flawed exercise. It could be a nucleus of a cell or a nucleus of an atom. And I was like, oh, good point. Because I'd already had another ambiguous one. And I said, but that isn't why the exercise is flawed. That's what gives the richness to the exercise. The exercise is not about getting the right answer. It is about understanding relationships, be able to argue them, and get a logical answer. And that's what this class is about. So, have a good weekend.